Thank you very much. As you know, my conflict of interest is that I'm not a pediatrician. So I'm here as a guest, but I've always been intrigued and motivated to try to improve technology for children because of this concept of transition gap between adult nephrology and pediatric nephrology. We have slow transfer of technological advances, and new technologies often are tested in adults only. There is lower technological knowledge in the area of pediatric nephrology and limited engagement of industry, mostly because of small size of the population and probably also small size of the business that can make somehow any type of approach to children a kind of orphan approach. So I think that we need to improve transition by cross-fertilizing between adult and pediatric nephrologists, work more together. Our knowledge may help each other, I think. We should support pediatric studies, share information, and also push charity and non-profit plans to get more and more engagement in developing this. I would like to start with this case that occurred in our hospital, 2.9 kilos. In 1979, patient with post-surgical acute renal failure, anuria and sepsis, nephrologist, me, was called to consult in pediatric surgical ICU. We did peritoneal dialysis catheter, and we did 35 milliliters per kilogram exchanges with 0 dwell time. Patient developed hyperglycemia, fluid overload. Patient deceased after 40 hours of treatment. I must say that we were pretty expert in peritoneal dialysis. As a matter of fact, probably our group was the leading group at that time. So in spite of that, we had problems dealing with PD in this patient. Infant dialysis in the early 80s was mostly treated with PD. PD performed with bottles or bags. You're probably too young to remember the bottles. We had gravimetric manner system. As a matter of fact, there was a very inaccurate measurement of ultrafiltration. The fluids were not so biocompatible, and frequent severe complications were present. Hemodialysis was almost not feasible. There was high demand and lack of technology. I must say that I published and I also did a patent on a specific called precision set system that was basically a Y set for peritoneal dialysis with small chambers to be applied to an adult bag in order to accurately measure the fluid volume for exchanges and the ultrafiltration. So there were need for alternatives, and we came from the experience of continuous arteriovenous hemofiltration in adults, where we had designed specific filters for the purpose of treating any type of patient independently on the blood pressure. And we did study the physiology of the artificial glomerulus, as it was called at the time, based on protein concentration, blood pressure, and the size of the catheter. So we move forward thanks to the collaboration with Amicon Corporation in Massachusetts first and Ireland subsequently, who developed a highly permeable membrane, and we started working at prototypes of filters that, as you can see here, were very, very, very small. One of the most important advancements, we convinced the company to create membranes with the expanded inner diameter of the fiber to reduce at the maximum the inner resistance of the filter. So the filter was looking more or less like this. So I present this case of 1980, 3.4 kilogram patient with anuria after abdominal surgery. Again, I was called to consult. Peritoneal dialysis was precluded due to large peritoneal breach, and we inserted cannulas surgically in brachial artery and jugular vein, and we did CAVH with the mini filter and modified blood lines with a blood flow of 8 to 11 milliliters per minute and fluid exchange of 1 milliliter per minute. This was the circuit that was developed at that time, and renal function recovered after 96 hours. I think that this picture has made a little bit the tour of the world, because that was the first patient ever treated with continuous arteriovenous hemofiltration. Then we subsequently published in Kidney International a series of 4 cases. I remember that I didn't want to publish 4 cases, but then I realized that in pediatric nephrology, 4 cases were not so small population, so it was quite curious for us. And then we had several other type of treatments, and we found differences in post-operative PaO2 and duration of mechanical ventilation in patients treated with hemofiltration. This is another baby treated at that time. We tried the bigger filter in case of hypercatabolism, but simply we found much simpler the application of countercurrent dialysate flow rate. It was not easy because the first filter only had 1 hole, so we had to create a new filter with 2 holes. And in fact, we described for the first time the so-called hemodiafiltration here in Vicenza. And you see from the blue line how much the uregeneration rate and urea appearance rate was controlled by the presence of countercurrent flow. So after that, we moved into a more professional design of the circuits, in a more professional design of different mini filters that more or less resemble the filters that are in part used today. And the most important thing was the incredible stability of mean arterial pressure and heart rate, especially thanks to a very nice volume measurement that we had thanks to the Equaline fluid balancing system, which was operated by 2 load cells from ultrafiltration to replacement solution. Vascular access, we used any type of vascular access helped by the surgeons. This was a case we treated in Mount Sinai Hospital in New York using umbilical vessels. And there was a specific guideline to insert catheters in the very early neonate for this use. But then suddenly we had the double lumen cannulas, and finally we could use pumps. However, as you know, from arteriovenous we moved to veno-venous with adoptive technology coming from the adult experience. This was part of the monitron machine, and this was a typical Christmas tree with many of the components added to the circuit, but not interfaced, so with high possibility of mistakes. Then technology developed with dedicated machines, and babies started to be treated with pediatric adaptations of circuits. Now, the interesting thing is that if you look at the percent of circulating blood volume compared to patient weight, you have to consider to try to keep the, let's say, the extracorporeal blood circuit very low. So we were strongly committed to try to design things that could be applied, especially in the small neonate, where all the applications of adult machines were considered off-label. You must have the right tool to do the task you are willing to accomplish, and size matters. So we wanted to create a dedicated technology. And the fact that AKI in the neonate was kind of an orphan disease prompted us to start thinking in terms of miniaturization. The history of medical devices was clear, moving in the field of pacemaker, for example. So we looked at what happened in the field of dialysis and computers. There is no comparison what the miniaturization occurred in the computer hardware compared to the dialysis hardware. And if you look at the machine, for example, in one company, more or less the machine kept the same design. So in terms of innovation, you may do incremental or radical. In this case, what you need is not an evolution, but is a complete revolution. And we took advantage of new sciences, microfluidics, nanotechnology, miniaturization in general, and inspiration from Galileo Galilei, who was the first proposing scaling down of machine and mechanical devices, Richard Feynman and Eric Drexler. I had the privilege to meet these people, the inventor of nanotechnology, crazy guy, but very, very inspiring. We were the first publishing on nanotechnology applied to dialysis and starting projects on wearable devices in conjunction with Victor Gura and independently in our own institute. Now, very often when you shoot at the moon, you miss it, but you may land among the stars. And this is exactly what happened to us when we started studying miniaturization. We felt that the best customer for miniaturization was certainly the neonate. So we did crowdfunding, team building, charity events, project design to create many events. You can see here hockey, sailing, skiing, and many others, including the concert. I'm sorry, but at the end, we collected 380,000 euro, which is a ridiculous sum for plans in the United States, but in Europe was sufficient to push in Mirandola, the technology pole, the evolution of what we started drawing from scratch, which was an initial design of a miniaturized machine. These were the first prototypes, and the dimension of these prototypes were meeting more or less the specification. You can see here the pencil and how we had designed the first prototype. The idea was to create something with a blood flow between 0 and 50, an ultrafiltration between 0 and 5 with an accuracy of 0.1 and a priming volume less than 30 milliliters. So we went to a more professional design. And finally, in 2008 and 10, we presented the first Carpe Diem machine and prototype with the specific pumps, filters, scales, and technology. We tested with our engineer, Francesco Garzotto, and Kim here, and with Dr. Ricci in our own institute. And this is the evolution towards the year until in 2013 we got the approval for human use. And this is how the machine looks like at the end, with several different characteristics. And actually, the Carpe Diem is displayed in the exhibition, so you may take a look to it. Now, the interesting thing, we had 3 different priming volumes from 3 different kits, and the smaller one was less than 30 milliliters, so it was exactly meeting our specification. Now, a case came at the end of August, the beginning of September, of a neonate with an enormous hematoma in the subgaleal position after complication during delivery. And the patient displayed enormous fluid overload. She displayed acute kidney injury. She started to receive a pharmacological approach, but she was totally anuric, and we placed a double lumen cannula for French. Actually, the cannula was very, very tiny. We consulted with Stu Goldstein and other friends, and the idea was that this probably would not have led anywhere, but we had no alternatives. This was the first baby ever treated with Carpe Diem, which has the characteristics of having very, very small shifts in pressure because of the size of the pumps compared to an adult pumps. So enabling the possibility to work with smaller devices. And in fact, we were able to reduce the fluid overload over time. Actually, the baby was treated for 25 days, and we were able to control creatinine in this baby. we were able to maintain hemoglobin and platelet count. The baby turned yellow at a certain moment because of liver disease. So not only we used the machine for dialysis, but we also used for blood exchange, using the 2 pumps for in and out blood. We also used that for single-pass albumin dialysis and plasma exchange. So we were quite creative in trying to save this baby. I would like to point out the color of the plasma coming out from the neonate. And these were the whole treatments we had done, including plasma exchange, but finally we could control both creatinine and bilirubin, and somehow come to extubation and to the recovery of renal function, which was at probably 85 to 90%, because she remained with a slight chronic kidney disease at the end. But this was the baby after returning to the hospital to visit Uncle Claudio, as they used to call this. And this was a real touching experience that made me write in this book that is now translated in English and in Chinese. And these were the SMS from Cincinnati. We got a 4 French lumen. You aren't going anywhere. It's working constant flow. It's impossible. Stu was amazed. The baby survives. It's a miracle, and the baby recovered. This will change the way we'll do medicine in the neonate with AKI. So after actually 30 years from the first publication of our CAVH first world treatment, we had another publication with the first Carpe Diem treatment that was published in the Lancet. What we call a smile worth 40 years in medicine. Thank you very much.